Welcome to the video tour of the Matyjaszewski Polymer Laboratory and office spaces as part of the Center for Macromolecular Engineering in the Mellon Institute at Carnegie Mellon University. Through the main entrance, both sides of the hallway belong to our workspaces with capacity for upwards of 25 researchers overall. On the left, we begin with chemical space. Fume hoods here are outfitted with Schlenk lines for performing polymer and small molecule syntheses. In between, we have an autoclave for glassware. We also have a centrifuge and a designated fume hood for handling hydrofluoric acid. Beginning in this middle portion is a larger room outfitted with shared instrumentation. This is our backup UV-Vis near-IR spectrophotometer, and here we have a triple detector for light scattering measurements, as well as our thermogravimetry station. Our primary UV-Vis near-IR spectrophotometer is used often, for example, in the analysis of transition metal complexes relevant to catalysis in controlled radical polymerization. These are our dynamic light scattering setups, both high throughput and single cuvette for size distribution analysis of nanoparticles and aggregates. We are outfitted with a glove box on site, multiple chemical refrigerators and freezers, and a solvent system. Moving past our freezers, we are out of the instrumentation room and back to chemical fume hoods. Currently, near one of our weighing stations, our electrochemistry station is set on a mobile cart. Crucial to our work are electrochemical analyses of transition metal complexes and electrochemically mediated polymerization itself. Before our next set of fume hoods, we have a rotary evaporation system. Often, one can see the use of photoreactors deployed to fume hoods for polymerizations. Moving past one of our solvent cabinets, we enter the original hallway, which we will return to the beginning on the other side. On the way, multiple research posters from our group are showcased. On the other side, we have office spaces for students and researchers. The next set of offices connects to the remaining instrumentation and chemical workspace. We have a deionized water station, followed by our lyophilizer, an automated flash chromatography system, and a vial rotary evaporator which can be used to evaporate small quantities with ease. Here is another fume hood, also for example employing a photoreactor setup. Neighboring this is a cold reactor and microwave reactor. To the right now is our gel permeation chromatography instrumentation room. Macromolecular samples can be dissolved in tetrahydrofuran and injected on the left chromatography setup, or an NN-dimethylformamide and injected on the right. On the insert is an aqueous GPC housed at another floor. The determination of molecular weights and their distributions are vital for analyzing controlled radical polymerization techniques. They are armed with refractive index, light scattering, and UV detectors. Moving out of the GPC room, we have more solvent cabinets and fume hood workspaces, also with an autoclave. Here is the third office space. And the fourth office space as well. As we near the end of the hallway, on the left is our chemical inventory. Across from one another then is our conference room. The 60 MHz benchtop NMR instrument is assigned here. We also use the conference room not only for group meetings, but also for discussing projects with one another and collaborators. Finally, we reach the head office spaces. This includes Dr. Kurt Olsen, Associate Director of the Center for Macromolecular Engineering, and our Professor Krzysztof Matyjaszewski.
Welcome to our lab and to the Center for Macromolecular Engineering. So far you have seen probably our lab already. We have nearly 20 hoods, and we have several very specialized equipment. We have GPC working in the water, working in DMF, in the THF with multiple detectors. We have some light scattering detectors, we have spectroscopy, we have also thermal analysis equipment and glove boxes and many other equipment, which sometimes we also share with other people from the department, also from other areas of CMU. So also you have seen probably offices. We have 4 offices for 25 students, undergrads, graduate students, postdocs, short or long-term visitors. And all of what we do is really macromolecular engineering. So we try to prepare polymers and macromolecules with precisely controlled structures. And in order to do that, we need to really focus on several aspects of research. So probably one of the most important thrusts is really how to control mechanisms of these reactions and how to prepare these catalysts which would be more robust, more selective, more active. We can use them at much lower concentrations, and under very benign conditions they can be regenerated by mild reducing agents like ascorbic acid, sometimes light or even electrical current. So the first group really is focused on this mechanism and the catalysis. The second subgroup, or mini group as we call it, will be involved predominantly in controlling polymeric architecture. So this will be making stars or making brushes, sometimes making rings even, making networks with precisely controlled structures. And sometimes also we try to depolymerize polymers back to monomers. The other two groups are probably a bit more focused on the applications and making polymers by combining inorganic substrates with nanoparticles or sometimes flat wafers with polymers and making them dramatically different lubrication properties, different dispersibility, different stabilities. And also we more recently look at the lithium metal batteries, how these concepts can be used to lithium metal batteries, which would be really working for many cycles, hundreds of thousands of cycles. And the final group is working on bioconjugates. So these bioconjugates is really a combination of nucleic acid, both DNA and RNA, with polymers, and also proteins with polymers. And essentially the idea is how we can sometimes make enzymatic reactions in organic solvents, how we can stabilize nucleic acids, how they can be delivered. This would include exosomes and some other cell-like nearly structures. So altogether, of course, these are very diversified topics, and for that we need to collaborate, and we collaborate within a group. Many students and postdocs, they have not one, but sometimes 4 or 5 projects running parallel, but also with people in the department and outside our department at mostly CIT, in material science or chemical engineering or biomedical engineering. And of course, we also collaborate with some other groups, not only at Carnegie Mellon, but at University of Pittsburgh, and it happens also that we collaborate with people from different parts, located in different parts in US, but also in Europe, in Italy, in Germany, in France, and also in Poland. And sometimes students really travel to these institutes and they learn different techniques in order to make it probably better and better, because I think science always requires a lot of the collaborative efforts. And I believe that every student in my group should be very deeply understanding what they do, better than I understand when they graduate, but also very broad by reaching to different areas, sometimes engineering, sometimes biomedical, environmental, energy related, and some others. So I think that you have already been exposed to many particular topics, but this was a brief overview, and I hope that you like our lab and our wonderful group of students, graduate students, undergrads, and also postdocs. Thank you.